Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is an event of the International Society for Extracellular Vesicles, and we're very pleased to be co-hosting today's event with Socrates. This is the Society for Extracellular Vesicle Studies in Singapore. And I'd like to introduce now the co-moderators of this event, Wei Seong and Minh, who are going to help to moderate and then also introduce our special speaker for today. So Minh, may I hand it over to you, please, to introduce the speaker.
Thank you so much, Ken. This is Minh from the National University of Singapore. It's my pleasure to introduce my colleague, Dr. Jiong-Wei Wang. He is currently an assistant professor at the Department of Surgery and Physiology. Previously, he graduated from the Leiden University Medical Center and did a postdoc training at the University of Utrecht and moved to National University of Singapore 7 or 8 years ago. So currently he also in the Cardiovascular Research Institute. His research interests focus on nanotheranostics with both synthetic and natural nanoparticles, including extracellular vesicles. So we're very happy to hear from him today about male extracellular vesicle. On to you, Jiong-Wei.
Okay, great. Thanks, Minh. Many thanks for the organizers to give me this opportunity. It's a really great opportunity for me to share our recent research. As I mentioned earlier, I'm a biologist in training, so I actually look at different nanoparticles from the angle of biomedical research. So we have been working on metabolism diseases, that's including cardiovascular disease and non-alcoholic fatty liver disease. I know that everyone sitting here are experts on extracellular vesicles. Actually, I do not need to give any introduction, but for the sake of our students and some newcomers in the field, so I'd like to give a very brief introduction of extracellular vesicles. So extracellular vesicles, in brief, it's also EV. And so the EVs, they are natural nanoparticles with a size of around 100 nanometer, and they could be secreted by all different type of cells. That's including all different immune cells or other resident cells as well. And those vesicles, they are present in all our body fluids, including the blood, urine, and also saliva. So in that way, these extracellular vesicles have been considered as a toolbox for biomarker discovery, that's why here, they have been used as a kind of a diagnosis tools for various diseases. And also because these extracellular vesicles, they are released under certain biological conditions and therefore contains various cellular components from their parent cells, including RNA, DNA, and proteins. And therefore they could function as a commuter between different cells, and therefore they have biological functions. And because of that, extracellular vesicles have been shown that have great potential of as a therapeutics for various diseases as well. And for that, these extracellular vesicles have been explored quite extensively initially, in the biomarker discovery and also as therapeutic reagents in the past. But in the recent years, extracellular vesicles also been exploited as a vehicle for drug delivery. And even nowadays, you have extracellular vesicles mimetics, or that's a different hybrid versions which have been presented before by Prof. Georgia Postori. And for today's talk, so I will focus more on the milk-derived extracellular vesicles. And also because these milk-derived extracellular vesicles are relatively less exploited compared with other extracellular vesicles. So we started to explore the intrinsic activities of these extracellular vesicles in the health and disease. So milk, and I think we are all very familiar with breastfed milk, with which we are grown up. So breastfed milk is our first natural food, which contains various nutrient components, and it's including different sugars, antibodies, hormones, and also multivitamins. As also, this milk also contains, the immunological components, as well as microbiomes or bacteria, which all help us to develop our guts and also the development of the gut microbiome. And in the recent years, it also found that this milk also contains extracellular vesicles, which is shown here, right? And this is the human human milk extracellular vesicles. You can see from the electron microscope, they are cup-shaped. That's why I really like Ken's background, with all the golden vesicles on the walls, right? And so this extracellular vesicles is not only present in human breast milk, but also in the milk from other animals. For instance, from the cow milk, which we drink daily, and also the goat milk, right? And the morphology looks very the same. And so when we started this project in Singapore, so I asked my students, just go to the supermarkets and pick up a few brands of cow milk, from the shelf, and then we isolate this extracellular vesicles from this, commercial fresh milk, and we noticed that, very interestingly, the content of extracellular vesicles are different among different brands. But for the conflict of interest issue, I won't I won't identify any brands here. But if you're interested, just drop me an email. I will tell you which brand of milk in Singapore contains more extracellular vesicles. But compared with other extracellular vesicles, you isolated from culture medium or even plasma or serum, and the extracellular vesicles isolation from raw milk is notoriously challenging. That's mainly because this raw milk are so rich in proteins and also other nutritional components. That make it very challenging. And there are several methodology for isolation milk EV have been reported. The first one, of course, is from Wang, and etc. They reported a methodology a few years back. They just take the similar methodology for isolating extracellular vesicles from other medium, right? Like the cell culture medium, plasma serum. So they combine sometimes, with isoclic, but mostly they just do not treat the milk and directly go to the ultracentrifuge. But this methodology, have contained get a lot of contaminants from the milk. That's because of this raw milk, contains so many different components, right? And the other methodology was developed a few years back, by Somia and colleagues. So they treat this raw milk first with a hydrochloric acid, and then in this way they could precipitate quite a lot of proteins from the milk, and from the supernatant they could isolate the extracellular EV with a relatively high abundance. So my colleague from China, Professor Huaxi Yi, and Professor Yi is a scientist in the food science field. So his group has been working on the milk and cheese, and this kind of milk products development for years, right? And they have quite long history for milk processing. And one of his talented students, and he's also sitting in the audience today, and he came up with the idea is why not use chemosen, right, to treat the milk first. It's a kind of a methodology used in the industry, to produce cheese, right? To precipitate the milk components and form the curd, right? Then you have the cheese production later, right? And in this way, this chemosen treatment could precipitate the most protein and also lipids, right, from the milk. And then you get a much better or clearer supernatant for milk isolation. And it shows indeed this methodology compared with the other two methodology reported earlier, they have a much better yield of nanoparticles and also much purer. If you see here, from all the three different methodologies, like what I mentioned earlier, that if you do not treat the milk with any compounds, and you get a lot of contaminants from this milk, right? That's why if you only use the protein contents as a parameter to measure how much of extracellular vesicle you isolated from the milk, it seems like the autocentrifuge methodology have large yield, right, compared to these two methodology. But actually, if you look at the purity by calculate the particle, the number of nanoparticles with the ratio of protein, right? It's kind of the way to evaluate the purity of the extracellular vesicles you isolated from this milk, right? If you look at the numbers, and the chemosen treatment method could produce much purer extracellular vesicles. And if you look at the diameters by the NTA methodology, and you could see that the extracellular vesicles isolated, after the chemosen treatment, they're around 100 nanometer, right? It's quite similar to the other nanoparticles, to the other extracellular vesicles. And in comparison, the milk EV isolated from the other two methodologies relatively larger. We assume that that's likely because this milk EV is also co-precipitated with some components from the milk. Then we move on and to do the RNA sequencing and also proteomics analysis, and to understand what's the content of this milk EV isolated from this cow milk. And from here, you see this, okay, they are very small, so you don't have to try to read all this type of, RNAs. They are all small RNAs, but to our surprise, more than 50% of the total small RNAs, identified from the milk EVs, they are actually microRNAs which are identifiable in the microRNA database. So this is very surprising, and it's also quite interesting as well, because we know that the microRNAs from extracellular vesicles, have been reported to have, all different therapeutic potentials, right? And similar to the milk EVs from cow milk, and the milk EVs from human breast milk also contains a large amount of microRNAs, which are known in the database. And with this top 40 microRNAs, we noticed that nearly 1/3 of those microRNAs are the same between cow milk EV and human milk EV. So that means these microRNAs in the milk-derived extracellular vesicles, they could be evolutionarily conserved. And further on, we went for the Kyoto Encyclopedia of Genes and Genome Analysis, pathway analysis. And we noticed that in among the top 40 microRNAs, most of them are closely associated with either epithelial tight junctions or the immunity or immunology pathways, right? And it's more like the same for the proteomic analysis. And we also noticed that, most of the proteins among the top 40, they are closely associated with tight junction and or, the immune regulations. With this in mind, and we have to, we are curious, how and where these milk EVs are from, right? In the cell culture, we know that they are either from this cell or that cell, but in the breast, and you have all different cell types from here. You can see here, right? This including this secretory cells, epithelial, right? And different fibroblasts, adipocytes, as well as smooth muscle cells, all these different cells here, these immune cells here as well. And apparently, these milk EVs, they are a kind of a cocktail. They have a different cellular region, and exactly for which cell specific region of the milk EVs have any specific function, it's still unclear. So the next question is, so when we have the milk EV and we take that orally, and where they go? And it's, most of the nutrients, what we think is, they could not pass the gastric acid, right, which is very acidic. But surprisingly, this milk EV, they could go through the tough gastric acid environment and reach the intestine, right, as shown here. And for the down, they may even go through the gut barrier and reach the bloodstream. And thereafter, they could, reach different remote organs along the circulation, blood circulation. And therefore, they have different functions, right? In the gut, they could regulate the gut microbiome and the gut immunity. And also in other organs, they could help to develop the immunity as well. And for us, so we also test it in animals. So basically, we just oral gavai of extracellular vesicles isolated from the milk, and then we label them. And after you can see that within 1 hour, and this milk EV could reach the intestine and even peaks at the small intestine. And they also reach the colon, of course, within 1 hour. But the concentration in the colon is peaked at 6 hours. And this milk EV could last quite long in the intestine system. So the question is, we know that in the infants, so this milk could reshape or help our body to build up gut microbiome. So our question is whether this also happens in the adults, right? So for that, to test that hypothesis, and we give that to the adult mice, and with the different doses of the milk EVs, and that's a daily for up to 2 months. It's really a lot of work for the students, but he's so keen to explore this field. And from here, we can appreciate that is after EV treatments, you can see that the abundances of various gut bacteria, they changed quite a lot, compared with the red color, which are, the control mice only received the serum by oral gavai, right? And these different doses have a different impacts on gut bacteria. For the sake of the time, I won't go for the details because I'm also not expert in gut microbiome. So it's just to illustrate there is very remarkable impact of the milk EV on the gut microbiome composition. Very interestingly, among those changed gut microbiome, there is a small subpopulation of gut bacteria. They are actually the main producers of the short chain fatty acids, right? And those are listed here from this, bacteria. And these short chain fatty acids mainly, this three, this acetate, propionate, and also the butyrate, right? And these short chain fatty acids, for the people who are not familiar, so they are actually mainly produced in our guts. With this gut bacteria to digest the dietary fibers, mainly from all the different food source, right? They digest the fibers, and then they produce this short chain fatty acid in the gut. And these short chain fatty acids, they are highly bioactive. They could go to the system from the blood and have a systemic effects, as well as they also function, locally in the gut, right? And it's known that these short chain fatty acids in the gut, they could act on the differentiation or proliferation of different T cells, including these T helper cells, regulatory T cells, which pop star nowadays, right? As well as the B cells, and by regulating these different immune cells, these short chain fatty acids, could have antibacterial, or anti-inflammatory effects in the gut. And follow this line, and so we went on to these animals and look at, some inflammatory or immune-related markers in the guts. As you seen here, this mucus 2 protein. It's a mucus protein in the mucosa layer, but it also has the immune regulatory function. And similar to the mucus, this milk EV treatment also have impacts, on other immune regulated factors like a GAT4 regulatory gamma, right? And also this immune adapter MYD88, right? And in addition to that, this milk EV treatment could also increase the secretion or production of the IgG in the gut, as well as in the circulation. Okay, so that means this short chain fatty acids really have the immune regulatory function also in the adult gut. And from here, you see this, we have 3 different doses of the milk EV, and it's a low dose, middle dose, and a high dose. And very interestingly, it's not like the more the better. And from here you can see this. And actually, that's the middle dose, which is 0.6 milligram per kg per day. And this dose of milk EV has the most significant effects on the gut immunity regulation. So the take-home message from here is the milk EV, isolated from the cow milk, they have the beneficial effects to reshape the gut immunity, gut bacteria, as well as to enhance the gut immunity in healthy animals. So our next question is whether this milk EV have any therapeutic effects in certain diseases. And the first disease, or also very obvious, choice is to look at inflamed guts, right? The one common gut inflamed gut disease is the inflammatory bowel disease, and here we just look at ulcerative colitis. Right? In this colitis, this due to genetic or environment factors or any other food poison, anything, right? So this could trigger the impairment of our gut barrier function, and therefore this gut bacteria and the gut bacteria products, right? They could go through this gut barrier and into our circulation. Therefore, they cause inflammation and also pain to the colon area. Yeah? That's they cause colitis in the human. And in the animals, very lucky enough, there is a very well accepted mouse model of colitis, which is induced by the dextrin disulfide sodium, right? With this kind of polysaccharides, and you could feed these animals, and thereafter this DSS could disrupt the gut microbiome, and they also disrupt the layer of the gut, right? Therefore, they could artificially induce the gut barrier dysfunction. So with this model, and we tested whether the milk EV have any therapeutic effects, and in that way, we firstly look at the symptoms of these colitis in animals. There is no surprise. If you look at this, is the control, the animals receive the serum only, and the model animals receive the DSS. Right? The body weight loss is very severe, and also the animals even have blood feces. And of course, this also causes the shortness of the colon. Right? So if you see, this is the control animals, and this is the DSS treatment, right? And this is the colon is much shorter, which are, all these symptoms are quite well known. And our interest is, whether milk EV oral administration could somehow, reduce these symptoms. And, very exciting, we could see from here, right? And this is golden lines. And I also like the golden colors. It's the golden lines, as well as this blue line, right? So as shown here, the different doses of milk, they have, various therapeutic effects, and they do could reduce the body weight loss from these animals, as well as they could somehow bring back this the colon length, right? So these are all the symptoms. And if you look at the histology level, and from here, this is the healthy colon, and here, this is the model colon. You see there's a lot of inflammation happening here in the colon layer. And after the treatment with milk EV, this is these symptoms are attenuated to a various extent. So that's, when we look at the inflammatory markers, and indeed, it's a milk EV treatment, you could reduce the production of inflammatory cytokines. And when we look into more details, we notice that this milk EV treatment could reduce this the production of inflammatory cytokines, mainly via the toll-like receptor and NF-kappa B signaling pathway, as well as the NLRP3 inflammatory pathways. At the cellular level, we mainly look at the T cells, right, which are quite important in inflammatory bowel disease development. Right? In these T cells, these Treg cells, which are positive for CD25 and FOXP3. And from here, you can see that, of course, after DSS treatment, this cell population also increased. But if you look at the milk EV treatments, so this T regular cells increased even further. And this is known in this model, right? And for the detailed analysis, we look at the subpopulation of T regular cells, which are producing the anti-inflammatory factor interleukin-10. If you look at this cell population, this after the milk EV treatment, this cell population in the colon significantly increased, right? And in the contrast, the T helper cells producing the pro-inflammatory cytokine interleukin-17A, this cell population are significantly increased in the colitis model column. And after milk EV treatment, this cell population is brought back nearly to the normal level. So in this case, we know that the milk EV could reduce the gut inflammation, both at molecular and also at the cellular levels. The next step is to look at the gut microbiome. It's very well known that in the inflammatory bowel disease, the gut microbiota is altered, right? And in this case, from here, this is the control groups. It's the colitis group disrupted by this DSS treatment, and this is the milk EV treatment group. And I won't go for the details, but from at all different levels of the gut microbiome, the milk EV could recover or restore the gut microbiome. It's especially at the family level in the blue color, as well as the genus level. And this is very special analysis. I even put a note here because I'm really not expert in the gut microbiome field, but my students knows much better than me. So he told me that this is just alpha diversity analysis to look at the abundance of gut bacteria from all different levels, and with all these kind of indexes, right? Child index and Sherman index. So you can look at the abundance or the compensation of this gut bacteria. And from this alpha diversity, you can see here, it's just examples. You can see, so there is a very clear trend for milk EV treatment to restore the gut microbiome. And also on the right, you can see this is better diversity analysis and look at to evaluate the overall similarity of gut bacteria. And this is green color with the healthy animals, and this is a purple color, in the colitis mice. And this is gut bacteria. The similarity are quite far, so that means they are not similar, to a large extent. And when you treat those mice with milk EV, and this similarity are brought back, they are in between. So that also shows you this milk EV could, restore the gut bacteria. So the take home message from here is, this milk EV could regulate the gut microbiota as well as gut immunity in the context of gut inflammation. But the question is, how? Oh, sorry. There is another analysis, and this is called collodium-based analysis. From here you can also see there are several beneficial gut microbiome. Their abundance is also recovered by the milk EV, right? Including this is a star beneficial gut bacteria called AKK. Right? I won't go to the details. Anyway, so our question is now is, so milk EV have effects on the gut microbiome as well as gut immunity, but how or is there any link or correlation between these two important parameters in the gut? So we did Spearman's ranked correlation analysis, and to our surprise, there is a very strong correlation between the gut microbiome and the different cytokine chemokines are produced in the colon. And this at the both family and genus level as the gut microbiome. So this concludes that very likely, and this milk EV could regulate the gut immunity or reduce the gut inflammation via this gut microbiota. And whether this is also via the mechanism, which is shown in the healthy animals via the short chain fatty acids, we don't know. We haven't studied that far yet, but that could be a very interesting question. Very interesting. So this for the gut, there is a gut attached junction. Yeah? So this from the beginning of my talk, and we analyzed this Milky Ways and noticed a large amount of the microRNA as well as the proteins. They are closely associated with immune regulation pathway as well as the TET junction protein. And therefore, we look at whether this Milky Way could protect the gut integrity. And the first thing we do is we started with epithelial cells, just in vitro, and we cultured the human epithelial cells, COG2 cells, right, in a trans well. And then we treat those cells with DSS and TNF-alpha, try to mimic the Collides situation in vivo. And from there on, we used this photometer to measure the trans-epithelial electric resistance. And from here, we can appreciate this electrical resistance is dramatically, decreased after this DSS and TNF-alpha treatment. In comparison, this milk EV treatment could rescue this loss of this electrical resistance, which also means that the milk EV treatment could, protect the integrity of these monolayer epithelial cell TET junctions. And from there, we also did fluorescence staining and look at some TET junctions, some key TET junctions. For instance, this occludin and ZO1, right? This is a control, and this green color is the staining of these TET junction proteins. And after the the inflammatory treatment, you see the loss, the quite loss of the TET junction proteins, and Milky EV could rescue to this TET junction to a certain extent. And this result is further confirmed by Western blots, which is shown here. And with this in vitro data in mind, so we moved to the in vivo. And for that, we generated another model. Well, it's a similar model, but it's more chronic inflammation, right? We feed the animals with DSS with certain intervals. Therefore, so the symptoms is less severe and the inflammation is more chronic. If you look at the TET junctions, they are highly associated with several key signaling pathways, like the AMPK, insulin-like growth factor 1 signaling pathways, as well as the glucagon-like peptide pathway. It's a hormone. And especially this AMPK pathway in the epithelium, right, is known to be activated by the interleukin 10, which is secreted by the regulatory T cells, which we presented at earlier. We did the Western blot analysis of these columns. We see here this is a phosphorylation of this AMPK, and if you look at the ratio, that's the milk EV treatment have a much higher phosphorylation of the AMPK. So that means the signaling pathway is enhanced, and it's similarly for the expression level of the other two regulatory pathways. And later we also did the quantification. We can see here, right? It's a milk EV treatment, and they could either at low or high doses, they both could restore the expression of this TET junction regulatory pathways. And accordingly, the expression level of the TET junctions, ZOO1, occludin, and also this junction or adhesion molecule 1 also recovered after milk EV treatments. And very interesting and also very inspiring is there was a very earlier report showed that in the non-alcoholic fatty liver disease patients, when they got the biopsies from the intestine, they say from here is duodenal, intestine, and they did some staining. Look at the TET junction protein ZOO1 expression in this intestine, and they noticed that the expression of these TET junctions in the patients is decreased, and they further give the patients some radioactive compounds called EDTA, right? They are to measure the integrity or the intestinal permeability, right? This component could be determined from the urine, and then they know this integrity, and from there they did a correlation, and they noticed that this intensity of the ZOO1 expression in the intestine is highly correlated with the intestinal permeability. But this is probably not a surprise, but the surprising finding is the intestinal permeability and the TET junctions in those patients are disrupted. So this is the key message, right? And so for us, we're also curious, whether this milk EV could repair the gut integrity in the fatty liver disease, which is more natural in a sense, right? So therefore, we have set up this MCD diet-induced NASH model, which is a very well accepted model in the field and also commonly used, right? And if from here, you have 3 groups: the normal group, the model group, and also the milk EV treatment groups here. Right? So basically, you have a few weeks to develop fatty liver disease. Afterwards, we feed the animals continuously for another 4 weeks, and then we look at the treatment, any treatment effects. And very exciting, we noticed that from the red color staining, the expression of the TET junction ZOO1, which was previously reported in the patients, right? This ZOO1 expression was dramatically decreased in the fatty liver diseased animal colon. This is the colon, and from in comparison, this is EV depleted supernatant. We use that as a kind of another control compared with the milk EV, and this has very minimal effects. And if you look at the treatment with the milk EV, the expression of the one is somehow restored in the guts. Right? And of course, this is also reflected by this increased inflammation in the gut, and also the this is the restore of the another TET junction called occludin, right, in the gut as well. And from there, this is just the TET junction at a protein level, and we also measured the intestine permeability by the fluorescent Dextrans, and from here you can see this red color in the fatty liver disease animals, and this intestinal permeability is significantly increased. And then this is the control, and this is milk EV treatment. This is just a very preliminary study, but we have shown that there is a clear trend for milk EV to reduce the intestinal permeability in fatty liver disease. And this is also evidenced by the concentration of NO in the plasma, it's just as an indicator of inflammation. So the take-home message so far is the intestine integrity is disrupted in MCD-induced fatty liver disease, and this disruption could be reduced by the milk EV treatments. And then we move on. There's in the recent years, there is a theory in the fatty liver disease is the exist of the gut-liver axis. So in brief, it means, in the healthy animals, right, this gut is intact, so they could control, the transfer of nutrients from the gut to the liver. But in the fatty liver disease or in the disease conditions, this permeability of the gut barrier is increased, and this causes uncontrolled transfer of gut bacteria as well as the pathogenic agents, through this gut barrier and got into the bloodstream, and they reach the liver, cause inflammatory injury to the liver, as well as the lipid accumulation in the liver. Therefore, they may promote the development of fatty liver disease. So this is kind of a theory so far. So our hypothesis is, can milk EV protect the liver from the fatty liver disease just for the sake of this gut-liver axis? Right? And for that, we did a proof of concept study here. And as you see, this is HE staining to look at the morphology of the liver. And you see, this is the healthy liver, very good morphology. And in comparison to the healthy liver, so the saline treatment, fatty liver, this MCD-induced fatty liver disease, as I just introduced earlier. And this saline treatment have, of course, it's a mock effect. And this is the EV depleted milk supernatant, also have, nearly no effect on this morphology improvement. But in contrast to these two controls, the treatment with oral delivered milk EVs have a very significant effect to improve the liver morphology here. And this is further confirmed by the lipid accumulation. Right? This is we did the oral red O staining, which is very classical methodology to stain the lipid and look at the lipid accumulation in the liver. Right? And from here, you see this is the healthy liver. It's hardly any neutral lipids there. But for this model groups, you see this red color that indicates in those liver there are large amounts of bad lipids accumulated in the liver, right? And these bad lipids are reduced after the milk EV treatments. This is the proof of concept, and although we assume there is gut liver effects, and our milk EV could treat from the liver, from the gut, and therefore have some therapeutic effects on the liver, but the exact mechanism is still on the way. So now I like to take the opportunity, to summarize the talk today. For the milk EV, isolated from the cow milk, they contain large amounts of immune-related microRNAs and proteins. And second, this milk EV, they could regulate the gut immunity and gut microbiota in both health and disease, right? And then this, of course, this milk EV, they could protect the gut barrier integrity and therefore benefit the liver against the fatty liver disease, likely via the gut-liver axis. And for the future studies, we still think the milk EVs are still very less exploited compared with other type of milk EVs. Right? So they need further characterization. And of course, the scalability is always an issue for large-scale study. Right? So this also needs further improved. And of course, we are very interested in the mechanism of those milk EVs, not only on the gut, but also how they exert the therapeutic potential on the remote organs, such as liver in this case, right, in both the health and also in the disease situations. And with this, I would like to thank all the grantors for the available grants to this study, and also the groups, especially these two PhD students, this Lim Junzhong and also Seong Zhang. It is a tremendous amount of work. And our great collaborator from China, Professor Yi from the Ocean University, China. He's a food scientist again. He initiated, he started this isolation methodology, for the milk EV derived vesicles, as well as thanks to his team in China, and also my collaborators in Singapore, including Prof. Georgia Pastorin, who is sitting here to support, and as well as my collaborator and also good friend, Prof. Storm from Utrecht. And of course, many thanks to your attention. I would like to take any questions for discussion. Thank you.
Yeah, thanks, Jiong-Wei. Yeah, so thank you very much for the very informative and interesting talk. Yeah, so as we are running very late to the end of the hour, so maybe we can just take one question. But I would actually encourage all of you that after this talk to actually send your questions to Jiong-Wei for further inquiry and understanding of his research. So if I may, I will just take one question. Okay, so from Martin, okay, who is joining us from very late at night from Europe. Okay, in the mouse model, okay, you were using the milk EV and compared to say9, but have you actually tested whole milk as a control, and were you able to claim an EV mediated effect instead of a milk mediated effect? So we will just have one question.
Okay, sorry for the long talk. Yeah, that's a very interesting question. So with this been asked several times. We didn't do that, but based on our chapter of the milk EV dose, and we noticed that to reach the therapeutic effects, what we reported here, we need the animals to drink quite large amounts of milk. That's more than half liters, which is not practical at all. And in that sense, we do expect either no or minimal effects just by drinking fresh milk, the whole milk. Sorry.
Yeah. So thank you.
Wei Seong, I just got word from the ISEV headquarters that we're allowed to go for another 10 minutes if we want to, and if people can. So we could go to a few more questions if you're up for it.
All right. Okay, that will
got
be
a
so
special dispensation.
Yeah, because I'm actually afraid of people from America and Europe who are like staying at very late odd hours with us here. Yeah, so we will just go for like overdrive of 10 minutes. All right. Okay, so yeah, so I think Jiong-Wei was explaining that I think using the EV actually allowed to actually concentrate the therapeutic EVs from the milk at a higher therapeutic dose, right? Mm-hmm. So going on, the next question we have from Minh, okay, who would always have very intriguing questions. So how many % of milk EVs are taken up by the intestinal cells, and how many % can get into the circulation? Is there any antibody appear against the cow milk EVs in the mice?
Wow, all these questions are really, really inspiring, but also very difficult to answer. We do not have any, answer for that. But roughly, we did some ex vivo experiments, with kind of gut system mimicking, from the mouse with, some enzyme, right? Also gastric acid, and all the way down to the colon. And we noticed that, this milk EV is around 6 to 70% of this milk EV could go through all these challenges and remain intact. How much % of this milk EV could really absorb it by these gut cells? We have no clue. And although here within 12 hours, we only observed these milk EVs in the gut, but there are some other reports as well as our own preliminary data. If you look, if you wait a little bit longer, let's say 24 hours later, you do appreciate some milk EV in the liver as well. Right? In the bloodstream, I don't know. We never tested, and also we have never tested any antibodies generated, against this milk EV. No, but that's a very interesting questions.
Thank you, Jiong-Wei.
Thanks, Minh.
Great talk.
Okay.
Well, we should, have an offline talk as well.
Yes, sure. Thank you.
All right. So another question from Martin. Could you speculate on the formulation in which milk EVs are applied? Can the formulation have an effect on the efficacy?
Which formulation that's referred to?
So it's asking about whether if the formulation of the milk could have actually effect on the milk EVs.
Oh, you mean the powder, this formula?
Yep.
There was an earlier study, very preliminary also. They showed that during the process of the milk formula, most of the EVs are actually destroyed. So whether those microRNAs or these proteins are still functional, that's not known.
All right. So further to his question, okay, regarding the size of chemosyn treated milk, have you done EM to test your hypothesis in other milk types?
The
EVs are associated to the particles. Yeah.
Yeah, we did that before. So for the EVs isolated, from chemosyn or from acid treatment or other, centrifuge, ultracentrifuge treatment, the morphology of the EVs are quite similar under the electron microscope. It's the only difference, I think, is the purity. Right? The purity and also the yield of these milk EVs, because with the chemosyn treatment, you could get, could remove the most of the protein and lipid from the milk, and it gets a much better, supernatant for milk EV process.
So from Emma, so what do you consider to be the mechanism of EV mediated modulation of the microbiota?
Very nice question. It's really, really great. I'm, quite grateful for those questions. I don't know. It's a short answer, so it needs a lot of, a
lot of further research that is needed.
Further research to really answer that. Yes. But.
So just another question from Minh. Okay, so are the EVs taken up by certain types of bacteria only?
It's another difficult question for me. The short answer is I also don't know. Based on the gut microbiome composition changes, they could be absorbed, or taken up by both so-called beneficial bacteria or pathogenic bacteria, because both populations, they changed, right? They changed somehow in the way of our favorites, and exactly mechanism we do not know, but there is some hint reported in the literature. It seems that the direct interaction between this milk EV and the gut bacteria, and they may cause or induce release of the bacteria exosomes. But this, the data are very, very preliminary, and I think there's a lot of hypos there because of the difficult, the challenging to study.
Yep. So I see Martin here. Martin, you have any questions?
Hello, thank you for your nice presentation. Greetings from the Netherlands here in the middle of the night. I think you answered almost all of my questions. Let me see which one I also had. So one of the things I was actually wondering is, with the chymosin treated milk EVs, have you ever tested in vitro their functionality? In any cellular assay?
Oh, that's a humor flag. Thank you so much for these nice questions. Yeah, so we did some in vitro assays, to look at the uptake. Indeed, this milk EV, they could uptake by this cell, by these epithelial cells, and as well as some immune cells in vitro.
Yeah, the reason why I'm asking is because their size is so completely different compared to the other EVs that you tested, that I was thinking this chymosin is a protease and maybe it is cleaving off proteins from the surface of the EVs, changing their size, but then also making it maybe harder to be taken up if it's done via a receptor, for instance.
Oh, actually, that seems unlikely because we in my lab have been working on different, the extracellular vesicles of a different region, right? Including mesenchymal stem cells and other, even some synthetic nanoparticles. But for the chymosin treated milk, and from there on we get to the milk EV, we don't see much difference compared with other vesicles. So I think that this chymosin treatment is unlikely to cleave the surface proteins on these EVs. Unless to certain specific receptors, we don't know. We did not really look into this kind of detail, but this is a very interesting question. Yeah, very interesting question.
Okay, and if I may shortly, so it's very interesting because we also look at bovine, for instance, with the acidification of milk EVs, that it completely changes their behavior compared to, for instance, EDTA treated milk. EDTA and citrate are often used as a means to clarify or clear caseins. So yeah, that's why I'm so curious about it. A lot of people use a single isolation method, and in milk there are so many EVs you can easily still get a few and characterize them, but there are really clear differences between the different treatments. So I think that's really important to keep in mind.
Yeah, absolutely. This is a very valid question. When we talk about milk EV populations, I absolutely agree. Each group lab is using one favorite methodology and to isolate the EV, and from there onwards they do all the different analysis. Whether I'm sure we are kind of biased because we probably only look at a certain subgroup of these extracellular vesicles, right? I know in a Utrecht group, you also look at the large and also small milk EVs, and for us, we look at it. It seems it's a kind of a mixture of both the small and the large EVs, and when we do the column, we also see some, you do be able to isolate the different EVs. But for us, whether the chymosin treatments will influence one group or the other group of milk EV, we have no clue. But yeah, if you are interested, we can talk further. Maybe we share some protocols and to explore or compare. That could be very interesting or very important in the field, right?
I agree. Thank you so much.
All right, thank you. Yep. So yeah, over to you, Ken.
Well, thank everybody. I'd like to thank everyone for participating today, and to our co-moderators, Seong and Minh. Thanks for your constant support of the EV Club. And I will also like to note that we will have another session with Socrates in 2 months, so in the beginning of May, and then again at the beginning of July. So we have talks already set up for those 2 months. And Jiong-Wei, it's been great to hear from you, to hear about this research, all of the ongoing developments, and we wish you all the best as you continue with this to advance the field of milk EVs. So thank you, everyone. We hope to see you again soon at another EV Club. And I want to wish you a great rest of the week, a great weekend as well. And see you soon. Bye now.
Thank you so
Thank
much.
you, everyone.